Thank you to the organisers for inviting me to speak today. I'm going to be presenting some work that's come from the labs of Alistair Matthew, who's in the audience over there. And I'm going to present two case studies looking at two monogenic variants in potassium channels that underlie disease phenotype and how we characterise those. So I don't think I put my mic on. There you go. Can you hear me now? So when you consider genetics and their involvement in disease, you can pretty much split this into two categories. There's the monogenic diseases. These are where you see a variation or a mutation in a single gene that causes a disease. These are often inherited diseases, and common forms of monogenic diseases that you might know are cystic fibrosis, sickle cell anemia, and Huntington's diseases. These are relatively rare diseases that occur with very small patient numbers involved. You can also think about the involvement of genetics in polygenic diseases. So these are associated with a number of variations or mutations in more than one gene, so a number of genes that cause the disease phenotype. They often involve many more patients, and examples of polygenic diseases include things like diabetes, heart disease, depression, and pain. Historically, these have been very difficult to study, but the availability of new technology, patient data in biobanks, and also the sequencing of the human genome has meant that now they're much more easier to study and allow us to understand the role of genetics in many more diseases. You may have heard Alistair's fantastic PhD student, Sam Bourne, talk about what his PhD studies in polygenic disease of pain and the involvement of multiple ion channels. But today I'm going to focus on our work from the lab on monogenic diseases. So what is the involvement in ion channels and rare diseases? Well, I think this beautiful figure from the ion channel library really illustrates that they have a huge involvement in disease. Over 215 ion channel subunits have been implicated in rare monogenic diseases, at least. It's quite difficult to see from this figure, but almost 25% of those involve potassium channels. And the main diseases that they represent are neurological diseases, developmental diseases, and cardiac diseases. So you won't be surprised to see a figure on potassium channels coming from Alistair's lab. But broadly speaking, there are 80 genes encoding potassium channels. These can be split into 3 or 4 families: the voltage-gated potassium families, the two-pore domain potassium family, and the inward rectifier potassium family, and the K-calcium channels. So my first case study is going to be looking at a voltage-gated potassium channel, KV2.1, which is encoded by the KCNB1 gene. So this is quite a basic schematic. Again, art's not my forte either. Showing an alpha subunit of the KV2.1 channel. It has 6 transmembrane domains, a pore loop, a voltage sensor on the 4th transmembrane domain. It has very long N and C terminus, and within the N terminus, you get the tetramerization domain there. And both the N and C terminus have been shown to be involved in inactivation and activation kinetics of the channel. 4 of these come together to form a homotetramer of KV2.1, but also for this particular channel, it can heterotetramize with other KV subunits, which I'll talk about a little bit later. And the KV2.1 is highly expressed in the brain, or mainly expressed in the brain, in cortical neurons and in cerebellar granule neurons, where it's involved in regulating neuronal excitability, but also in some other organs such as the heart and pancreas and in the retina. You'll see that there's a number of clinical variants, quite a few for this particular channel, that have been published in the literature. I've just put them on this figure to show where they sit on the channel. You can see that there are a number of clinical variants throughout the whole of the channel, on the transmembrane domains, on the intracellular and extracellular loops, in the pore region, and in the N and C terminus. The one that I'm going to focus on today, I should probably point, is in the N terminus, this one here, and I'll just tell you about that. So this is Jack. He's 6 years old in this picture. He was born after a normal pregnancy to two unaffected parents, a 30-year-old mother and 39-year-old father. After 18 months, he was referred to the pediatric unit of UCLA Hospital due to the identification of global development delays. And there he underwent exome sequencing and was diagnosed with a likely pathogenic de novo heterozygous mutation in the KCNB1 gene. This was consistent with his clinical features of global development delays, hypotonia, muscle laxity, and speech delays. And sorry, I forgot to say, but all of those mutations in the previous slide, I'll just go back actually because it's important. Many of these mutations have been functionally tested, and most of them report a loss of function, but most of them report intellectual disability, global development delays, and behavioral issues. And all the ones highlighted in blue report seizures, and those highlighted in yellow report no seizures. So very much, if you're going to get a mutation in this channel, you're going to have developmental delays, some form of intellectual disability, and the potential to have seizures. So Jack, he had obvious development delays at 18 months, hypotonia, muscle activity, and obvious speech delays, but he did not have any seizures, unlike many of the other mutations in KV2.1. And so the mutation that they found after exome sequencing was in the N-terminus of the channel, proximal to the tetramization domain, where a proline at position 17 had been converted to a threonine. So this is Jack now at 6 years old. At 18 months old, he had these conditions. At 6 months old now, 8 years old, he has, fortunately for him, relatively normal tone, strength and growth compared to his peers. He's still currently without seizures, which is fantastic. But the main thing that he suffers with now is speech delays, strabismus, a short attention span, and many of these KV2.1 mutations in diseases, monogenic diseases, have showed that many of them suffer with ADHD or ASD. And his current treatment for his condition just involves really intensive speech therapy and some form of physiotherapy. So we were approached by his clinician, John Graham, who will crop up again a bit later. He asked us to characterize this particular mutation. And so we created a construct of the mutation in KV2.1, and we expressed this in a mammalian cell line, a modified HEC293 cell line. And what we did was use a step voltage protocol where we increased 10 millivolt step from -80 to +60 millivolts and measured the current that was elicited from the channels. And we did this for cells just expressing GFP and those expressing just homozygous KV2.1 and homozygous a mutation of KV2.1. And you can see that we saw huge currents running through the mutated channel at all voltages, and we averaged the current we saw at +50 and normalized that to capacitance, and you can see the difference in between the wild type and the mutated channel. So we can confirm that we felt that this was a gain-of-function mutation that Jack had. So I mentioned earlier that the KV2.1 channels can heteromerize with other KV subunits, and one of those is KV9.3. This is termed a silent subunit. Homotetramers of this channel are not functional and remain retained in the ER, but when it combines with 2.1 channels, it can regulate the function of these channels, and they are a bit more selectively expressed, but found in the cerebral arterial myocytes and in the cortex. So one of the most prominent effects of the KV9.3 silent subunit is to shift the steady state inactivation curve for KV2.1 to the left. And this means that less channels are available for activation around the resting membrane potential of -70 millivolts. So because this mutation was one of very few that sit in the N-terminus. We wanted to investigate this a bit further because we know that the N1 and the N-terminus and C-terminus have been implicated in the activation and activation kinetics of this channel. So to do this, we co-expressed the mutant construct and the wild-type construct with the KV9.3 channel. And what we saw was, as we might expect for KV 2.1 wild-type, was that very few channels were now available to activate over all the voltages. But for the mutant channel, we saw that nothing had really changed and that you could activate the channel over, the same amount of channels were available for activation over all the voltages tested. So we wanted to look at this in a little bit more detail. And to do this, we utilized a protocol where we did a long preconditioning pre-pulse step at different voltages from -100 to +10. This allowed us to reach, this was for 30 seconds, and allowed us to reach a steady state in activation before we applied the test voltage of +50 millivolts. And we were able to measure the amount of current that was given at +50 after each of these steps and normalize this current against what we saw at -90 and plot them on this graph. So for KV 2.0 alone, you can see that at -40 millivolts, you still have many channels that can be activated. And so you see much larger current coming through it. But as expected, when you express it with the 9.3, co-express it, that curve is shifted to the left. And now at -40 millivolts, less channels are available for activation. And this is shown quite nicely when you overlay the curves here on this graph with the pink and the orange or yellow. So we did the same thing for the mutant construct, and what we found was, like for the wild type, at -40, many channels were available for activation. However, when you expressed it with the mutant channel, with the 9.3 subunit, nothing has really changed. So the same amount of channels was available for activation, and you can see this here also. So more channels, yeah. So we also tried to see if there was any pharmacological alteration in this channel, and I'm going to pronounce this wrong, I think, but we looked at a selective inhibitor of KV 2.1, a tarantula toxin, giangitoxin. I'll call it GXTX. And it's been shown before to be quite selective inhibitor of KV 2.1. And you can see at 10 nanomolar and 100 nanomolar, you see quite a nice inhibitory effect of the channel. For the mutant construct, although we saw no real effect at 10 nanomolar, when you went to a much higher concentration of 100 nanomolar, then you could bring in an inhibition. So the channel could still be inhibited, but at much higher concentrations. So to summarize, this is a gain-of-function mutation, which both for the channel itself, but also when expressed with silent subunit 9.3, it shifts the steady state inactivation of both the homo and the heteromeric channels to the right, and also has slightly altered pharmacological response, which might have implications for therapy in these patients. And I think we can take from it that the gain-of-function phenotype is probably responsible for the lack of seizures that has been shown in JAK so far, but doesn't obviously compensate for the clinical symptoms such as development delay, cognitive impairment, and speech delay. So next, I'm going to talk about a monogenic disease that is found in the two-pore domain potassium channels and the KCNK9 gene that encodes for TAS3. So this is a schematic of a two-pore domain potassium channel. Unlike the voltage-gated, it has 4 transmembrane domains and 2 pore loops, and a very large M1P loop that sits extracellularly, quite a short N-terminus and a long C-terminus. And only 2 of these dimerize together to form a functional channel, and each of these pore loops on each alpha subunit contribute to a single channel pore, a single pore in the channel. They're expressed highly in the brain, but also all over the body. TAS3 is expressed all over the body and have involvement again in regulating neuronal excitability. So in 2008, Veale et al. Reported a new mutation in TAS3, a glycine to an arginine mutation that sat in the transmembrane 4 domain of the channel. And then it existed in several members of an Arab Israeli family, including the mother, and they called it Veale mental retardation syndrome because many of the affected individuals had moderate to severe intellectual disability, but I will call it Veale syndrome for now, and you'll see that we change it as we go along the story. Also, they had severe feeding difficulties. So again, very similar to what we saw with the KV2.1 monogenic disease, suffer from generalized hypotonia and weakness of proximal muscles. Many of them had dysmorphic facial features, so a very elongated face, a short philtrum, arched eyebrows, and often with a cleft palate. I should say that TAS3 is quite a rare ion channel that is paternally imprinted. So there's very few genes that do this. But what it means is that the paternal allele is silenced and only the maternal allele of these channels is expressed. So Veale, when they functionally tested it, Veale found that this mutation was non-functional when they expressed it in oocytes. And I'm not quite sure why we decided to look at this mutation. Another story, and I can't quite remember why, but we decided to look at it ourselves a few years later, and I expressed this in a human cell line. And what I found was that we found some very small but functional outward current at minus 40 millivolts, which was a lot less than the wild type, but was present, shown there. So we did some functional studies on this channel and found that the G protein coupled receptor inhibition of these channels, which highly regulates 2-pore domain potassium channels, was attenuated in the mutant compared to wild type. And at the time, we've been working with some activators of potassium channels, 2-pore domain potassium channels, not TAS channels, but what we decided to do was apply one of these, a phenamate compound, flufaminic acid. And what we found was that we were able to partially restore current through this mutation. And so the implication was that potentially these phenamate compounds could be a potential therapeutic benefit to patients suffering with this disease. So we thought that was it, and we moved on. But at the same time as we published the paper, we were contacted by the clinician that contacted us before. This is his patient, Preston Kelly. Here, he's about 19 months old. He was born, again, to healthy parents. He was... But from the very early onset, there was obvious problems. He couldn't feed properly. He had dysmorphic facial features, and he stayed in hospital for a few months. At 4 months, whole exome sequencing identified him with the G236R mutation. And being parents of a child with a syndrome that's being diagnosed, they went and searched the internet and found our paper. And Melissa contacted us, and her clinician contacted us, asking us about our work on FFA. In the States, FFA is not available, but mefenamic acid, which is an alternative phenamate, is FDA approved for use in the States. And so it was decided at 14 months that Preston would start on a regime of MFA. And he initially showed very good promise on this compound. He was much more responsive. He started to roll at 19 months. He was much more vocal. And when he was off, he was 3 weeks on the regime and 1 week off. On the week off, he was very unresponsive, lethargic, and they felt he was better on it. But this didn't last indefinitely, and now he's not currently on the medication. A case study of him was published along with 3 other patients at that time, but it was very clear that Preston was on the severe end of, despite having the same mutation as the other patients, he was at the severe end of all the symptoms for this disease, and he's still unable to walk unaided, and he's 9 years old today. So we thought, again, that would be the end of it, but we were contacted by by the Mayo Clinic, by Margot Cousins and Eric Klee, who had a patient with a mutation in the C-terminus, just at the end of the 4th transmembrane domain, and methionine to a threonine mutation, and they asked us to functionally characterize this. The patient had many of the symptoms of the other patients that suffered from Veale, and so we did. We looked at that. I think what surprised us for that mutation was instead of a loss of function, which you see with the G236R, we saw a huge gain of function with this channel. So we've got now gain of function mutations in TAS3 that both cause the symptoms of this disease. And from that point on, we were bombarded, because of these papers that are now out in the public domain, we were bombarded by clinicians and patients with mutations, all of them displaying symptoms of this disease. And most of them, not just G236R, but we had a hot spot over here as well of this arginine here. So we spent a lot of time characterizing all of these mutations. Took about 5 years in total. And finally, we got this published. But you'll see here the G236R, that current density is down, the regulation by extracellular pH and muscarinic regulation is down. But for most of these mutations, we saw a gain of function. So just to summarize, so you have gain of function mutations or loss of function, all are causing the same disease, which is interesting. But the regulation of most of these channels was attenuated or impaired for things like G protein coupled receptor regulation and pH. And the severity of the disease phenotype can vary significantly between individuals, even those expressing the same mutation. And I think it shows that therapeutic approaches won't need to be individualized for patients because they won't always work for one. I'd just like to say a very small thing about Melissa, who is Preston's mum. She's a huge advocate for what they could now call KCNK9 imprinting syndrome. I think they wanted to move away from a disease that was named by the people that discovered it. And now they had the reason to do that because there were many more mutations that they were able to pull on to do this. And we're really fortunate to have been invited in July to the first patient meeting of the KCNK9 imprinting syndrome. And we'll be meeting with patients that we've studied and talking to them, collecting more data about these families, and we'll be and also we'll be meeting some of their clinicians. So I'd just like to thank Alistair, who's allowed me the opportunity to work on these projects, some of the PhD students that were involved in this study, and our external collaborators. Thank you.
Okay, thanks, Emma. Maybe get time for 1 or 2 quick questions, given the time. Some questions up here. There's one down here.
Hi. So this a common thing is that the gain and loss of function mutations seem to have a similar phenotype. But I just wondered, normally you're just showing your voltage clamp experiments. Have you looked at, say, current clamp? No. What's the impact on the action potential firing?
No, those experiments for these. No, sorry.
Here. Here. Yeah. Oh, okay.
Oh, sorry.
I'll go. Okay. So my question is regarding your first case, whether you have exclude the possibility that the mutation what it's doing is to not allowing KB9.3 to bind to the channel, because we have data with Wangi toxin, and we know that the selectivity is not the same when you have homotetrameric KB2 channels than when you have channels with other silent subunits.
Yeah.
So maybe
It's po-
that's the
yeah,
case.
It's possible, but we haven't got the evidence to show that.
You could do some experiment with we did antibodies in the pipette or looking at
Yeah,
immunoprecipitation or something like that. But maybe what is going on is that you cannot get this heterotetrameric
Yes, well,
channel.
because the mutations in the close to the tetramization domain, it's
Yes.
possible that it's affecting that. happening.
Okay.
Yeah, a bit related question. So these patients are all heterozygous for the mutation. So did you try, for instance, for the KV channel, co-express, wild-type, mutant,
Yeah,
and the
and
silent
we
ones? What
did.
do you get then?
Yeah, we did for the KB2.1, we expressed it with homozygous, and we saw exactly the same. There was huge gain of function. So
It's a dominant...
yes, it's a dominant mutation.
Okay, thank you.
Okay, maybe we're just going to stop there in the interest of time and move on to the last speaker. So Emma, thanks again for your talk. Thank you.